In this lecture, we continue our discussion of receptors, and now we're moving away, of course, from nuclear receptors as activators of transcription of drug metabolizing enzymes, talking more now about receptors involved in signal transduction, and specifically some of the therapies that we use to target those. Now, of course, we're going to talk more about the pharmacogenetics and or genomics of those processes more than we will on the specifics of those drugs and the pharmacology of those drugs. And I hope that you will be willing to follow me through this discussion, because it's going to take us a winding way through some literature to finally get us down to the biologicals. And we're going to begin mostly looking at colorectal cancer. And I want to begin with the inhibitors of replication. And these are the camptothectins that I'm referring to here. And when we talk about the polymorphisms that we see in those, in the drug metabolizing enzymes and other enzymes involved in these disease therapies, what we see is a failure. And what do we do when those irinotecan and the other camptothectins fail? We see the development of a new series of inhibitors, and these are going to be tyrosine kinase inhibitors. Gleevec is going to be our particularly interesting example. I'm going to tell you a little bit about that, but also talk about some other and second generation tyrosine kinase inhibitors. But we're also going to see where they fall short and how we turn to other, or in combination with other therapies, treat these colorectal cancer tumors. And this is where the biologicals come in. And in turn, those have each of these, the pharmacogenetics relates at each level. So we're going to hopefully use Gleevec as one example, and we're going to turn to the standard inhibitors of epidermal growth factor, Erbitux and Vectibix, better known as cetuximab and panitumumab, respectively. And I'm also going to refer to other tyrosine kinase receptor based signal transduction mechanisms or pathways, and I'm not going to describe them here, but just so you know that we'll be touching on several of these. So yeah, a winding way of getting from nuclear receptors to the discussion of biologicals and the pharmacogenetics of the biological treatments for colorectal cancer. So, camptothectins. Now, there's a little bit of history here. The camptothectins, of course, are built from or derived from a series of natural products searches in the 60s. Camptothecin itself, the initial test molecules derived from a tree from China. I'll just write that down for you. Tree, China. There's a whole issue of intellectual property and where profits go that's sort of buried in there that we're not going to go to. What I really want to discuss, before leaving that notion, though, of the natural products, let's remind ourselves that Taxol and Paclitaxel are drugs that are also derived from natural products, and modification, optimization of drugs initially derived from natural products. So irinotecan is the drug that we're going to focus on. Decades down the line after the original isolation of these natural products, is a proisomerase type 1 inhibitor. So it is involved in cleavage and reannealing of DNA during replication. Now, in your mind, this is a distinct class of chemotherapies, but in your mind you might lump it together sort of with the metabolic drugs for chemotherapeutic agents. I'm thinking of methotrexate, which arrests the folate cycle. In any case, a means of shutting down a key process in the cell, in this case by inhibition of topoisomerase 1, that then arrests the development of the cell and leads to the destruction of the tumor. So this replication being the key element of the cellular processes that we're working on arresting. So that's what these drugs do, and these are still important treatments for colorectal and other cancers. And in this little snippet that I've stolen from Staker et al. 2002, we see a key piece of the mechanism of this kind of drug. And here, irinotecan is the crystal structure of irinotecan bound to DNA, and we see in the apo form we have the standard active site, the normal configuration of the cell of the protein, and here on the right we see irinotecan intercalated into the DNA. And I won't go into the details of the biochemistry, but you can easily imagine how that's going to reduce the efficiency with which the enzyme is going to seal up those breakages that it introduces into the DNA to unwind it. So it's an intercalator. It's a really clear structure here. The cool thing for us is that in the topoisomerase 1 gene, there exist polymorphisms that result in these mutations, and I've highlighted a few. Well, the authors have highlighted a few here. These are mutations that result in a tumor that is refractory to treatment. So mutations result in resistance. Interesting. Just a really interesting thing. But the point being, of course, that we spent a long time and lots of decades developing this disease therapy, and we still have difficulties implementing it. And those difficulties arise, of course, from a molecular basis. And the question that we have to ask is where do we go from there? But we're not quite ready for that. So let's skip ahead to yes, pharmacokinetics, right? So taking us back to week 1, it all falls together. I hope you see that here's CPT11, sort of the initial trade name for development name for irinotecan. You can see that there's CYP activation pathways, which are of course going to lead to its excretion. But there's also another drug metabolizing enzyme that we haven't talked much about. This is carboxyesterase, right? And this one also involved in the activation of this drug. So the active form of irinotecan is, of course, SN38, and let me just point out that, as I said, camptothecin is the core structure here, and there's a lot of variations on this theme. We're only focusing on one here. This is the camptothecin core structure. Oops, I can't spell. Core structure. And here's the irinotecan bit that makes it so interesting and unique and powerful. Now, for us, so you certainly could imagine that there are issues that might arise from signal SNPs, polymorphisms in the CYPs, for example, and also carboxyesterase. But the key difficulties with treatment by this drug arise from UGT1A1 and a particular set of substitutions that we'll talk about. This enzyme, of course, is responsible for the glucuronidation of the drug, which of course results in its excretion. Now, this enzyme, which we remember from those early lectures, is also involved in terms of endogenous metabolism with modifying bile salt and bilirubin. So heme degradation is our endogenous pathway that we're talking about, and of course the individuals that are polymorphic or carry the wrong version of UGT1A1, which we'll discuss in the next slide, also have disorders that don't come into our discussion here, but also have other metabolic disorders. In any case, it's the conversion of SN38 to SN38G by UGT1A1 that results in its efficient excretion from the body. Failure to do so, of course, is going to result in side effects. And so in this sort of cartoon, I have a couple of pieces of data to show you. But first of all, let me say that the side effects are important and severe. So a common side effect is diarrhea, but also more relevant to the current discussion is neutropenia, which I'm not going to discuss in detail what neutropenia is, but it basically relates to the immunosuppressive nature of this drug and the death of neutrophils. So as we'll see, I think in the subsequent slide, your neutrophil complement is one way to represent how well you're responding to the treatment. In any case, it's kind of fun to show this genetic structure of UGT1A1. I say here dizzyingly complex gene structure and transcription. Well, the bottom line being, without spending too much on a digression here, is that there are a series of exons, and the final transcript contains one of these unique exons. So and then 4 of the others. So there are 9, and I should just note that there's a couple of pseudogenes, so those don't count. But what we wind up with generally is 9 variants. But and that could be one of those initial exons followed by the common exons, and of those, 3' splicing yields 27 distinct mRNAs. So you can imagine that the organization and regulation of this gene is under some pretty tight control. Again, that's really an aside. What I really want to talk about, however, is the untranslated region of this gene. And what I'm showing you in this table here is just a series of polymorphisms, right? We remember from our earlier discussions that this star number identifies what the change in the genetic structure is, or what the polymorphism is. In the case of the efficacy of irinotecan, it all hinges on these TA repeats that fall before the translated region of the gene. So forgetting entirely in this discussion what the complexity of regulation of this gene at the level of transcript composition, it's these TA repeats that determine how well one responds to the drug. And but the bottom line, of course, is that TA7 equals poor expression of UGT1A1, and therefore, of course, poor excretion of the drug, and hyperbilirubinemia, and the neurological phenotype of neutropenia, and of course, in addition to those serious problems, of course, irinotecan resistance. So this, and of course, there are others as well. We see other polymorphisms that we're not going to go into here as well, but you can see here that they're also resulting similarly in decreased expression of the gene, and of course, diminished functionality, and a poor metabolizer phenotype. We're highlighting this one, and this one gets all the attention because UGT1A1 28 is the most common mutation, or the most common polymorphism that we see resulting in irinotecan resistance. Otherwise, I'm just showing you this fun data. Here's a gel showing you the ability of the technical expertise of the author in finding the difference between TA7 and the wild type TA6. So TA6 versus TA7. And in here, of course, those of you that are used to looking at these sequencing readouts, you can see here's our wild type set of TAs, and then wild type plus 1 TA. So you can just see there how the authors went about identifying what these polymorphisms were that were causing the resistance to the drug. Now, more on that, looking at going from mapping to actual data. There's a couple of different papers here, some key data points that are worth repeating, that are I find really fun to look at. First, again, reminding you that we're just focusing on the polymorphisms that in UGT1A1's untranslated region that result in resistance to our camptothecin topoisomerase 1 inhibitor, irinotecan. And what we see here is so this is a metabolic marker. Remember, I told you that the mutations in this gene also affect the excretion of the natural substrates for of this molecule, and so we're looking at bilirubin levels here. And of course, so an elevated bilirubin level means that we're not properly modifying the bilirubin for excretion. And basically, what we're looking at here is the level of bilirubin in each of these groups of patients. Those that have the wild type, those that have the increased number of this is sorry. Let me go back and say that this is referring, I guess, to the heterozygosity of the individual. So we have 2 copies of 6TA. Here we have a mixture of 6 and 7. Here we have a mixture of 6 and 8. And here we the homozygous 7/7 TA gives us, this looks terrible scatter, of course, but we look for our statistical analysis to say that the bilirubin levels of these individuals are elevated. Even before we start thinking about irinotecan, the bilirubin levels of these in these individuals are elevated relative to the homozygous wild type. And so in terms of looking for biomarkers and thinking about how we might screen for patients that might be resistant to this disorder, or understanding resistance when we find it, we might imagine looking not only or merely at the genetics of the individual, but also at the phenotype of the individual. That is, how well they're excreting their endogenous waste products. Now, skipping ahead though to the xenobiotic effects, so let's think of this as endogenous effects. Here's the xenobiotic, measuring the xenobiotic effect, and here the A and C nadir. Okay, I hope you're all very literate and understand what the word nadir means. If you don't, there's a new word for you. But basically, we're looking at the trough of neutrophil concentration here. Now, what does that mean? So this is when neutrophils drop to a minimum. This again is the immunosuppressive nature of this drug, irinotecan, and we see that the inability of excretion of the drug leads to a loss of neutrophils in the blood. And how do we measure that? Well, again, here is our homozygous 7TA. Oh, let me write it the proper way. TA7 homozygote. And we see that again. There's okay. So let me point out. Huge amount of scatter here, and it's always fun when you're reading these articles to think about the issues that we have with them, and invariably in this kind of a study, it's just going to be the nature of the numbers, few patients and we try to draw meaningful information from a relatively small cohort. But here, what we see is overall a very noticeable difference. I'm asking you to look at the scale here, right? How is this plot scaled? Logarithmic scale. And what we see, of course, is that there's a significantly lower neutrophil count in those individuals that have the poor excretion genotype. Okay, now, of course, that's all great. We have biomarkers, but what does this tell us about the efficacy of the drug? And what we see is that, I should mention that I think we've seen these plots a little bit in the past, but this is a Kaplan-Meier plot, so-called Kaplan-Meier plot. These individuals were instrumental in pushing through statistical analyses of disease progression. This was back in the 60s. But these are standard plots for looking at disease progression in cancers. And what we simply see here is, in the solid line, the survival rate, that is, how quickly are we losing patients over what amount of time, over the course of the study for the wild type upstream sequence of UGT1A1. And we see that significant differences, right? Here, let's pay attention to here, at the end of the study, significant differences between the survival of those wild types and those with the TA7. The extra TA repeat in the upstream region, the 3' region. So in this case, we can clearly see that that with the wild type is going to, in the presence of irinotecan, is going to result in a higher survivability. So this is, if you're a patient, this is important to you. Why are we going to take this drug? And why does it matter what our UGT1A1 genotype is? I should also mention that I think these when we see an asterisk or a hash mark here, we're talking about someone dropped out of the study and we don't necessarily know the reason for that. So we don't need to get into the statistical analysis of that, but in any case, I think it's intuitively obvious what we get from this kind of analysis. All right, so let's stop with the camptothectins there and irinotecan there, and simply say that there are lots of ways to affect lots of factors, let's say, that affect the efficacy of irinotecan. Most importantly, your UGT1A1 genotype. But of course, it's also important to look at these other biomarkers as well. Now, even though irinotecan is an important chemotherapy for colorectal cancer, issues such as the one we're describing here make it very important to develop alternatives. And so in a subsequent lecture, I'm going to go into begin to talk about some of those alternatives, in particular small molecule inhibitors of tyrosine kinase receptors.